I'm going to talk to you today about advancements in pharmacogenomic research, and I'm going to describe a streamlined workflow for sequencing and analysis of genes associated with drug metabolism pathways. My name is Fiona Hyland. I'm the Director of Bioinformatics in the Clinical Sequencing Division at Thermo Fisher. So, pharmacogenomics is an important research area. There are many genes that affect the metabolism of psychotropic, cancer, pain, and cardiovascular drugs, among other diseases. And specifically, we focus on the ADME process involving drug absorption, drug distribution, drug metabolism, and drug elimination. The most important family of genes involved in these processes are genes involved in the cytochrome P450 pathway, including CYP2D6, CYP2C19, CYP2C9, and others. These genes metabolize more than 80% of common drugs. And when we consider how an individual is going to metabolize or absorb or eliminate these drugs, we consider factors such as, are there variants in any of these genes which may affect these processes? Is there copy number variation in these genes? And what does the expression of these genes look like? And the goal of this kind of research is to understand the genotype of the individual with respect to genes that affect these pathways and processes, with a goal of maximizing drug efficacy and minimizing adverse effects. So we wanted to develop a panel for next generation sequencing that addresses this need. Some of the challenges we faced are homology between the genes. As I mentioned, there are a lot of CYP genes that are highly homologous with each other, and they can be difficult to distinguish from each other. And so this was the primary challenge that we faced. We also wanted to develop a panel for the detection of copy number variation. We wanted to ensure that we had a high call rate, high sensitivity, high specificity, and we could develop a panel with acceptable cost. And our solution was to develop an ion AmpliSeq panel containing targeted content. This is the Ion AmpliSeq Pharmacogenomics Research Panel. This panel contains 119 amplicons in one pool. It also has 9 sample ID amplicons. It includes targets in 40 different genes with 136 total SNP and indel target. And in addition, 1 copy number target, which is identifying copy number state of the CYP2D6 gene. And you can see on the right all of the genes that are included in the panel. These include genes relevant to metabolizing drugs involved in pain, psychiatric disorders, and oncology, among others. More specifically, this is the number of targets in each of the genes in the panel. You can see that CYP2D6 covers more than 20 different targets in CYP2D6 gene. CYP1A2 has 15 targets. And then there's another 4 genes with more than 10 targets, going down to quite a number of genes that just have a single target. In addition, we include amplicons from the sample ID panel, comprising 9 autosomal primer pairs. These were selected to have high minor allele frequency across populations, to be in Hardy-Weinberg equilibrium, to be unlinked to each other, not to be in genes, not to have any function, and to have high heterozygosity. And in addition, we include amplicons designed to identify gender. And so we report out on a little genetic barcode, if you will, containing the sex of the individual and these 9 letters corresponding to the genotype of that individual. This allows us to distinguish between thousands of different individuals. And so this is very useful for tracking purposes. Our development process included 92 cell lines with known genotypes for the genes involved. We had buccal swab samples that contained known challenging genotypes and known copy number state for CYP2D6. And we collected buccal swab samples from multiple sites from volunteers. And we characterized those samples using TaqMan open array, TaqMan copy number assays, and where necessary, Sanger sequencing. And the development process included redesign of the assay. It included a lot of bioinformatics work to optimize the analysis of this panel. One big advantage we have with AmpliSeq panels is that we can design the primers in a very precise way to enrich just for the gene that we're interested in and not for other genes in the same gene family that are very similar. And so we developed an analysis solution to take advantage of this knowledge of the primer context, adjust the amplicon map quality value on an amplicon by amplicon basis, and in addition, to account for amplicon strand bias. And we developed a Torrent Suite plugin that incorporates all of this bioinformatics optimization, and that also includes quality control, QC analysis, data analysis of the sample, filtering, exporting, and reporting. We support automation, and we offer 8-plex on a 314 chip, up to 96-plex on a 318 chip. And we worked with a collaborator for independent verification in an orthogonal laboratory. Some of the challenges we face when looking at a panel for evaluating these gene families are that if one does a naive analysis, one can mistake a homozygous variant for a heterozygous variant. For instance, this is an example of a variant in CYP2B6, which we know to be AG from an orthogonal technology. However, if analyzed in a very naive way, this might appear to be an AA homozygous, and it's because of the some of the ambiguities of mapping between these two genes, CYP2B6 and CYP2B7P1, that have high homology in this region. But because of the advanced bioinformatics algorithms that we developed, we're able to disambiguate these two genes, map the reads to the correct gene, and correctly identify that this is, in fact, an AG heterozygote. We want to make sure that we have accurate identification of this genotype because it's associated with a relevant phenotype. And so, for example, before we do optimization, if we use the naive approach, we found we only had 51% concordance with some orthogonal data. After optimization, where we take into account the known primer sequences and the known primer locations, and we adjust the bioinformatics analysis, we end up with much higher concordance. So I've told you about some of the work that we've done to improve the detection of SNPs, insertions, and deletions. There's another variant type that's important, and that's copy number variation, specifically for the CYP2D6 gene. CYP2D6 is probably the single most important gene for pharmacogenomics. It metabolizes many commonly used and important drugs, including tamoxifen, codeine, and many others. And there's known copy number variation in human populations. People might have 2 copies of the gene, the normal diploid state, in which case they're an extensive metabolizer. But it's also possible for people to have 0 copies because they have a homozygous deletion. That's the CYP2D6*55 variant, and those people are poor metabolizers of these drugs. They may have one functional copy, the star 5 deletion, in which case they'll be likely an intermediate metabolizer. But they may even have 3 copies, in which case they're an ultra-rapid metabolizer. However, it's also possible for people to have a certain number of copies of the gene, but they may not have functional copies. So there is a variant that exists in human populations, which is an exon 9 copy number. It's the result of a gene conversion event. We call this the star 36 allele. And if that exon 9 variant is present, that makes one of the copies non-functional. In that case, an individual may have the following genotype, where they have 3 copies of CYP2D6 gene, but only 2 of them are functional because one of them exhibits the exon 9 CNV, this star 36 allele. And in that case, rather than having 3 functional copies and being an ultra-rapid metabolizer, this individual only has 2 functional copies, and they are an extensive metabolizer. And so we wanted to detect not only the gene level copy number, but also the exon 9 level copy number, so that we could correctly characterize the metabolism status of this gene. First of all, we identified some samples with known null copy number, 0 copy number. Those samples are pretty easy to identify. So there wasn't much of a challenge with identifying individuals with a null genotype, null copy number. A larger challenge came in distinguishing between individuals with 1, 2, and 3 copies and the star 36 allele. So we developed a novel method for detecting copy number for CYP2D6. We normalized with the median of all of the non-CYP2D6 autosomal amplicon read counts. We developed a Gaussian mixture model with EM clustering, and we cluster in log 2 count space. We implement some fairly rigorous QC, so that if the QC, if the quality value is too low, we determine that that sample has a no call, meaning it has an unknown copy number, and we won't call it if we're not confident that we can make the correct call. And we developed this and confirmed it on both our Croyel samples, cell lines, and also buccal swabs. So we did this both for the copy number of the entire gene, of the entire CYP2D6 gene. And we also repeated this to develop a method to detect the copy number of the exon 9. And what you can see here, if you look at the samples in blue, when we look at gene level copy number, they have 3 copies. There's dark blue and there's pale blue. But then when we look at the exon 9, the dark blue ones show that they only have 2 copies of exon 9, and the pale blue ones show that they have 3 copies of exon 9. And so this is how we can distinguish individuals with the star 36 allele with only 2 functional copies of the gene. All right, so I've talked to you about how we developed the assay and the analysis solution. Now let me show you some results. So this is results that we got from our V&V process. We measured 304 cell line specimens on the PGM system, 336 buccal swab specimens for a total of 640 specimens, and an additional 320 specimens on the S5 sequencing system. We made a total of 86,000 calls on PGM, another 43,000 on S5. The number of no calls on the PGM was 25, with 3 incorrect calls. We actually didn't make any no calls, and we had no incorrect calls on the S5. So our genotype call rate was 99.97% or 100%, and the genotype accuracy was above 99.99% or 100% on the S5. Turning to the results of our copy number variation. we sequenced samples that had copy number of 0, 1, 2, and 3. And we included individuals with 3 functional copies of exon 9 and with only 2 functional copies of exon 9. And you can see here the results for each of these samples. I've shown here the no calls and the number of true positives. We actually did not in our V&V results. And so you can see that the call rate on the S5, 100%, on the PGM was 99.8%. The exon 9 call rate for copy number was variously 96 and 97%. So we were able to demonstrate that we've developed an assay and an analysis solution that has high call rate, high sensitivity, and high specificity, both for SNPs and indels, and also for copy number variation. We partnered with an independent lab. They ran 48 samples, and they determined that the genotype sensitivity and specificity for those samples with known variation was 100%, and their call rate was 100%. And for copy number, they had 100% sensitivity, 100% specificity, and a 98% call rate for genes, 100% call rate for exon 9. The analysis solution can be accessed using a Torrent Suite plugin. This includes optimized mapping and variant calling. It includes hotspot calls, and it includes CYP2D6 whole gene copy number variation calling, and also CYP2D6 exon 9 star 36 allele copy number detection. The plugin can export results that are compatible with downstream analysis solutions. This is software to help interpret this data. There's an internal piece of software that's also compatible with TACMAN assays. And in that situation, the customer provides the hotspot to star allele mapping. It doesn't have any clinical interpretation. It is free. Or there are some paid solutions. One I'd like to point out is translational software. This is a very comprehensive software solution. It is on it paid. It does cost money, but this will provide a full clinical interpretation of the genotypes of the individual, describing the star allele status of the individual, and also will have an extensive report describing the impact of these genotypes on the metabolism of various drugs. So I've told you about the pharmacogenomics panel that we developed and launched. Another thing I want to point out is that this panel can be customized. So you may look at the genes and the targets involved that are included in the panel. And you may decide that you would actually like to add some preferred research targets that you're interested, or you feel that some of the genes that are present are not very useful or relevant or appropriate for your research, and you'd like to remove those targets. The panel can be extensively customized. You can add essentially any number of targets in any genes. You can remove targets that are undesired, and you'll want to update the hotspot file to add additional targets. The one constraint on the panel is that we do need to keep all of the CYP2D6 amplicons unchanged in order to enable CYP2D6 copy number variation detection. In addition, we need the panel to remain as a single pool in order to do this, again, to do the CYP2D9 copy number variation detection. So if you want to do CYP2D6 copy number detection in this panel, those are the constraints. If you don't, the constraints are gone. And if you customize the panel in this way, you retain the ability to use the pharmacogenomics plugin with your customized panel. You retain the ability to detect CYP2D6 copy number status, including both the whole gene and the exon 9 copy number. and you can continue to use the reporting software. And you can contact your field team in order to get more information about this customization if you desire. So in conclusion, we developed and launched the Ion AmpliSeq Pharmacogenomics Research Panel and its matching Torrent Suite software plugin. It was verified on the Ion PGX system and the Ion S5 sequencing system and by our collaborators at Gentris. It uses buccal swab. It can take as little as 10 nanograms of input DNA. So it shares the excellent property of AmpliSeq that it is very robust to small amounts of input DNA. It has high demonstrated sensitivity, specificity, and call rate. It supports detection and reporting of single nucleotide variations, insertions, deletions, CYP2D6 whole gene copy number, and star 36, as well as the sample ID. And interpretation and reporting software are available, and this is a customizable panel. Thank you very much for your attention. Hope I'll take any questions.